right, so I want to make sure that you can see the shared screen on your side. Can you please let me know if you do? All right, great. All right, so I want to first go over the final review for our lab exam coming up. I don't remember what exactly was the exam date. Can you remind me, please? I believe it's going to be next week, I believe. November 9th, yes. All right, so we're going to start with our lab review, and then we're going to be moving on to cover the new material from the blood chapter. So first, I want to go over the main diagrams. And as what we're used to, we're going to be rating them from 5 stars to 1 star. For it to be ideal when you are studying the material, for you to know how to prioritize your work. So looking at this first diagram, I would rate this diagram is a 4 stars out of 5, so it's relatively of high priority. What is shown here on the diagram, we have the different components of my urinary system. If you remember, the urinary system is formed by the kidneys. You have right and left kidneys. And this is where the filtration of the blood gonna be taking place. For the urine that got filtered, extracted out of the blood to be traveling down through those muscular tubes. Those are my ureters. And the urine will be accumulated within this muscular bag located at the inside of the pelvic cavity. Located inside the pelvic cavity, this is going to be my urinary bladder. Once the urine fills the urinary bladder, if you remember, starts to contract. And as it contracts, it squeezes out the urine to travel out through the urethra. So we're looking here at the main components of my urinary system in this diagram. First of them is going to be my kidneys, right and left. Then I have the ureters, urinary bladder, and urethra. If you remember, all of the components of the urinary system, along with the major vessels, the large veins that you have in the abdominal cavity here, this is your inferior vena cava, and the large arteries that you have traveling in your abdominal cavities, this is the abdominal aorta. All of this is located behind my peritoneal cavity. So I'm not located along with the other organs, I'm located retroperitoneal. What I mean by retroperitoneal, behind the peritoneal cavity. So all the rest of the abdominal organs are kept within the membrane, within the peritoneal membrane. This doesn't include my kidneys and major vessels. You see on here, the 2 arteries that are direct branches from the abdominal aorta that will go to supply the blood to my kidneys. Those are my renal arteries. And remember, the blood coming out from my kidneys gonna be drained through the renal veins to drain directly in my inferior vena cava, and it doesn't share with other abdominal organs in their venous drainage as what we've seen back when we discussed the venous system. Remember, most of the abdominal organs will drain the deoxygenated blood to travel back to the liver before it gets into the systemic circulation for the liver to process everything coming out from those organs. Here, it's not the case for my kidneys. Kidneys will drain directly through my renal veins to my inferior vena cava. And remember, The abdominal aorta splits into two terminal branches, one on the right, one on the left, and those are my right common iliac and left common iliac. And for the inferior vena cava, it receives the blood coming up from my pelvic cavity and from my lower limbs through my right and left common iliac veins. So here, two branches of the abdominal aorta, right and left common iliac arteries. Here, two branches of my inferior vena cava. Those are my right and left common iliac veins. Again, this diagram is a relatively of high importance for us. It's a 4 stars diagram. A relatively lower importance than the previous diagram is this diagram showing a transverse section of the abdominal cavity. I will rate this as a 3 stars diagram. And if you see on here, this is my peritoneum. This was including my small intestine, my large intestine, my stomach, my liver, my pancreas. All those abdominal organs are kept within the peritoneal cavity. So I removed all those all those abdominal organs kept in the peritoneal cavity. So I will have here the peritoneal cavity after I removed the organs. Located behind my peritoneal cavity are going to be, as we mentioned in the previous slide, the different components of the urinary system along with my major vessels. One of the common wrong answers I usually see on the exam, people identifying this major artery traveling in front of the vertebra as renal artery. It's not my renal artery, it is my aorta. It is a large, huge artery traveling in the abdominal cavity. And some people will identify this as inferior vena, as renal vein, but it's not the renal vein. It is the major vein traveling in the abdominal cavity. This is my inferior vena cava. So those are two of the most common wrong answers I usually see on the exam related to this slide. The two branches coming out from my abdominal aorta go into my kidneys. Those are my renal arteries. And the two branches coming out from the kidneys to drain in my inferior vena cava. And those are going to be my renal veins. The kidney is held in place by multiple factors. One of them is the fat surrounding it. This is my renal fat, and I have a capsule like any other abdominal organ. It is surrounded by a capsule. This is my renal fibrous capsule. All right. Any questions? Any questions? All right.
So going back, this is Hannah. Going back to the perinatal, do we need to know the
peritoneal cavity is this one?
Yes. Do we need to know the irregular dense CT underneath the fibrous capsule?
No.
Okay.
All right. What are the important landmarks that you need to be able to identify on this diagram? Is that this is called the peritoneal cavity, not the abdominal cavity. This is my inferior vena cava. This is my abdominal aorta. Those are my renal veins. Those are my renal arteries. Of course,
Okay,
the kidneys. And you don't have to worry about the perirenal fat or the fibrous capsule or any of that. All right, so moving on to another diagram, a highly, highly important diagram. I would rate this as a 10 stars diagram out of 5. Ten stars out of 5. It has to be on your test. What do I mean by 10 stars again? It has to be on your test. It's not negotiable. If you remember, this is a transverse section of the kidney. What I can see on here, I can see an outer layer of the kidney. This outer layer, all this part, all of it is my renal cortex. All that outer layer of the kidney is my renal cortex. And all that internal layer is my renal medulla. So you have two layers forming the kidney. An outer layer, this is the renal cortex. An inner layer, this is your renal medulla. At the junction between the cortex and medulla, we've got triangular-shaped structures. You remember, they are not triangles because they have depth. So there is a third dimension that I can't see from this view. So this is actually a pyramid. So what do we call the pyramids of the kidney? Something related to the kidney is renal. So we're gonna call this pyramid is my renal pyramid. Renal pyramid. And if you look, you will find a pointed apex of each of those renal pyramids. This is where the urine gonna be traveling to be drained after it gets formed from the tip of this pyramid. What do we call the tip of a pyramid? We call this is my papilla of the pyramid. The papilla or papilla of the pyramid is the pointed apex of my pyramid. The urine that gets formed gets conducted down to small ducts. So what is the definition of those small ducts? You need to remember this. The small ducts are the ones that will be receiving the urine directly from the apex of a renal pyramid. The ones that collect the urine out of the tip of the renal pyramid, those are my minor calyces, regardless of the size. So if I get closer on here, and we're looking at this one, you see, it's huge. Would I consider this one a major calyx? It's large. What do you think? Yes or no?
No.
Would you? No. Why not? Because simply it is directly connected to the apex of my pyramid. If I am directly connected to the apex of the pyramid, I am a minor calyx, regardless of the size of the duct. So what's going to happen to the urine after it gets through my minor calyces? It's going to be conducted to large ducts, like this one here, like this one. Those are what we refer to as my major calyces. So by definition, what is the major calyx? It is a duct that receives the urine from multiple smaller ducts. So if I am collecting the urine from multiple smaller ducts, I am a major calyx, not a minor anymore. So you see this one, major. It collects urine from multiple ducts. This one here is a major calyx. This one is a major calyx. This one is a major calyx. This one is a major calyx. If I'm connected to more than one duct that I'm draining the urine from, I'm going to be considered a major calyx. regardless of the size. So you see this one here, it's directly receiving the urine from the tip of a pyramid. So it's not a major calyx, although it might be even larger than this one. All right, so what's going to happen to the urine after it gets conducted from the minor calyces to the major calyces? The major calyces will end up conducting that urine towards a funnel-shaped tube. This is my funnel on here. It funnels the urine towards this muscular tube. This funnel is what we refer to as my renal pelvis. Then that urine gonna be conducted down to the muscular tube here. This is my ureter. So again, every single marking on this diagram is of great importance and you need to be able to identify. So what are we looking here at again? Renal cortex, renal medulla, renal pyramids, the papilla of the pyramid, minor calyces. If I'm connected directly to the tip of a pyramid, I'm a minor calyx. If I'm connected to multiple ducts, I am a major calyx. And the major calyces will end up draining the urine into this funnel here. This is my renal pelvis. And from the funnel, I will conduct the urine down in a form of droplets through my ureter. And as we've seen in the earlier diagram, the ureters will conduct those urine droplets towards the urinary bladder until it expands and then contracts to release that urine out through the urethra. Moving on to another must be on your test diagram, 10 stars diagram. So if I'm looking on here, this is my functional unit of the kidney. This is my nephron. And if you remember, any nephron is formed of two main components. I have kidney tubules, and associated to those kidney tubules, I've got blood vessels, capillaries. So kidney tubules. First of them is this cup-shaped tube. This is my Bowman's capsule or the glomerular capsule. Please remember both names for your test. So glomerular capsule or Bowman's capsule. And embedded within that Bowman's capsule, you've got a collection of blood vessels associated with it. This is my glomerulus. So remember, at that point in my nephron, this is where the very first step of the urine formation takes place, which is my filtration. So fluids, along with solutes, gonna be passing from your blood in your glomerular capillaries to be filtered into the Bowman's capsule. So what creates this push is mainly the hydrostatic pressure of the blood. And one of the main contributing factors that affect that high pressure within the glomerular capillaries is the difference in the diameter between the vessels that brings the blood into the glomerular capillaries and the vessels that drains the blood out from the glomerular capillaries. This is my afferent arteriole, and this is my efferent arteriole. So more blood gets in than what I'm draining out. And as a result, I create a state of high pressure within my glomerular capillaries. This is what drives the filtration taking place here. So the fluid that I extracted from the blood is what we call the filtrate. The filtrate will do what? Will keep moving along my kidney tubules. So it keeps moving first along my proximal convoluted tubules, and then travels down in a part of the tube that looks like a loop. We call this loop on here is my loop of Henle. Notice the part of the loop that descends, that is coming down, is connected to your proximal convoluted tubules. So if you see two parts of the loop of Henle, look back, where am I connected to? Am I connected to the set of tubules that will get me back to the Bowman's capsule? Or am I connected to a set of tubules that will take me towards my collecting ducts? To know which one is going down, which one is going up. So the one going down, carrying the fluid down, this is the one connected to my proximal convoluted tubules. The one that will be conducting the filtrate up is the one connected to my distal convoluted tubules. Another key difference between this part of the loop and this part, the descending limb and the ascending limb of the loop of Henle, is the difference in the diameter, the thickness of the limb of the loop of Henle. So if I'm looking on here, I see most of the length of the descending limb is formed by the thin segment. And most of the length of my ascending limb of the loop of Henle is formed by my thick segment. Again, connected to the thick segment of the loop of Henle or the ascending limb, you've got my distal convoluted tubules that will carry on the remainder of the filtrate after you complete the reabsorption, after you complete the secretion. It becomes the urine that will be collected from many nephrons through my collecting duct. So every single marking on here is highly, highly important. It must be on your test. This diagram gets some of you frustrated. Want to go ahead and break it down for you. So what is shown on this diagram is my two types of nephrons. As we've learned, the outer layer of my kidney is my renal cortex. The inner layer of the kidney is my renal medulla. 1 million nephrons are going to be located within each kidney. So those are microscopic structures. All of them are kept in by one way or another within my cortex, like the proximal convoluted tubule, the distal convoluted tubule, the Bowman's capsule, along with the glomerulus. All this from all the million nephrons is kept in my cortex, in the outermost layer of my kidney. Here. But we've noticed that some of the nephrons that are located within my cortex far from the medulla. So this is the line that separates my cortex on top and the medulla at the bottom. We call this is my corticomedullary junction. Corticomedullary junction is simply the junction between the cortex on top and the medulla at the bottom. So, looking here at the first type of nephrons, we've seen that it's pushed up into the cortex. It's mainly located in the cortex, up away from my medulla, down here. And it will have a short loop of Henle. So we called those nephrons, because they are pushed up deep into the cortex, we call them cortical nephrons. And we've noticed that there is another type of nephrons that is still mainly located in the cortex. So the Bowman's capsule, the proximal and distal convoluted tubules, all that is located still in the cortex. But it sits close to the medulla, very close to the junction between the cortex and medulla. So we called it juxtamedullary. Juxta means close to. Medulla, medullary means to the medulla. So those are my juxtamedullary nephrons. And they are characterized, those that sit close to the medulla are characterized by having long loops of Henle. So the loop of Henle, as you see on here, it travels deep into the medulla, surrounded by a large set of vessels that travel straight. So we called it the straight vessel. We call it vasa recta. So all your nephrons are kept in the cortex, but 15% of them are in the cortex, but they are close to the medulla. Those are characterized by having long loops of Henle. And what are those long loops of Henle serving for? They serve for you to pull water from the filtrate back to your blood. This will end up causing the urine that you are forming to be concentrated. As you keep pulling the water out, the urine becomes more concentrated, becomes darker in color. The solute content is going to be higher. How about this set of vessels that are
Can you
branches?
repeat that one more time?
All right, so if I'm looking here, this is my juxtamedullary nephron. They are characterized by having long loops of Henle. And why do I need to have such nephron? Why not all of them are cortical? I have those juxtamedullary nephrons that will have long loops of Henle surrounded by vasa recta to allow me, if I needed, to pull water from the filtrate back to my blood. And as I pull the water from the urine or the filtrate that will become the urine back to my blood, if you put a cup of coffee in the microwave, what's going to happen to the water? It evaporates. So you are pulling the water out. And as you pull more water out, what's going to happen to the concentration of the coffee? It becomes more concentrated. So what the juxtamedullary nephrons are serving mainly for, along with the long loops of Henle and the vasa recta, is that they are going to be responsible to create concentrated urine if I need it. So in some cases, I won't need it. If I'm drinking enough water, I will not need to pull that amount of water back to my blood. Right? If I have excess water. But if I need it, because I'm not drinking enough water, what's going to happen? I will pull this water back to my blood, preventing its loss out in the urine. Does this make sense?
Yes, sir. And then you said something about sodium. So the sodium quantity is going to be higher if that's the case?
Solute content, not sodium. Solute means all ions, all waste products. Solute content, not sodium. Do you get this?
Yes, sir. Thank you.
All right, so this is a part that gets some of you confused. Which one is my proximal? Which one is distal? So, on the exam, you can zoom in. So you're going to have similar diagram, and you can zoom in. And when you can zoom in, you can simply follow the tube. So if I put my marking in here, if this is your label, what do you think this is? Is this proximal or distal? Without looking at the identification, at the label. How would I know? Or this? Or this here? Or this? So let's try this one. All right, can you answer whether you can see a respond in the chat box by identifying the number as whether it's proximal or distal convoluted tubule. And I will zoom out here a little. All right, so let's check out this together. All right, so what am I looking here at? This is my loop of Henle, right? So if you can remind me which part the descending or ascending limb is mainly formed by the thin segment. Descending here. So the urine is coming through down here. And it will go up through my thick segments that forms most of the length of my ascending limb. So this is something that you need to be able to recall. So if you are connected to the descending limb, so this is number 2 on here. What do you think you're on? If I'm connected to the descending, so 2 is going to be what? Proximal, exactly. 2 going to be proximal. How about this one? Number 1, it's connected to the thick segment, ascending. So this is going to be my distal. So 1 is going to be distal. How did I figure this out? By looking at the loop of Henle. Does this make sense so far? All right. So what if I'm looking at here? This tube on here. Let me follow it. It travels below the Bowman's capsule and keeps going to join my distal, my collecting duct. Which part of the convoluted tubules is gonna be connected to the distal, to the collecting ducts? How do you think? Is it proximal or distal that is being connected to the collecting duct? Distal. So number 3 here is gonna be distal. And how about number 4? This one here. So follow it. I will follow it back. See where it will take me. You see, it's connected to my Bowman's capsule. So you follow here. Cut like this. You end up at the Bowman's capsule. So this is going to be proximal convoluted tubule. So number 4 is going to be proximal. Exactly. You see, so how would I figure this out? Whether this part of the convoluted tubules is proximal or whether it is distal, I need to be following the tube to see where it gets connected. If it's connecting to my Bowman's capsule or to my descending limb, I am proximal. If I'm connected to my ascending limb or my collecting ducts, this is going to be distal convoluted tubule. And if you remember, what did we call the capillaries around my proximal and distal convoluted tubules? Those are capillaries around the tubules. So we're going to call them peritubular capillaries. And where do they come from? Remember, those are capillaries from my efferent arteriole. Efferent arteriole. So you get afferent arteriole bringing the blood in, and the efferent arteriole carrying the blood out from the glomerulus after the filtration gets conducted. Any questions? Any questions?
So we need to know the so do we need to know the afferent and the efferent and like what they do as
Yes.
far as like the capillaries?
Mhm.
Okay.
All right. So if I'm looking here at the corticomedullary junction, I have this vessel in red. This is the artery that gets the blood delivered to the small structures, the glomeruli. This is gonna be my arcuate artery. It forms an arc separating my cortex from the medulla, arcuate artery. And then it gives rise to branches that travel deeper into the cortex, penetrating the cortex upwards. We call those are my cortical radiate artery. So if I'm drawing something like this, you tell me what does am I drawing? What does this look like? Can you see what I'm drawing here? Sun, right? So this is exactly what we're looking for. So this one, this vessel on here, is like this arc. So we call it arcuate. And I have radiated rays radiating out from the arcuate artery. So we're gonna call those are my cortical radiate arteries. And this is the origin of my afferent arteriole. So the afferent arteriole is connected to my cortical radiate arteries that are coming up from my arcuate artery. And how about the efferent arteriole? Efferent arteriole it's going to be connecting on one side to the glomerulus and to the other side to the peritubular capillaries. All good so far? Any questions? Any questions? All right, so moving on here. A relatively important diagram. What I want you to focus on in this diagram are the 3 steps of the urine formation. If you remember, what are the 3 steps? You filter the fluid along with the solutes from your glomeruli into the Bowman's capsule. What do we call this step in the urine formation? We call it glomerular filtration. What do we call it? Glomerular, because it takes place in the glomerulus. And it's a filtration process. It's not tubular filtration. It's not tubular whatever. Right? It is my glomerular filtration. Don't just settle with filtration. All right? You need to remember the whole name here. Glomerular filtration. What if I filtered stuff that I still need, like glucose or amino acids or whatever? If I filtered stuff that I still need, I have to capture them back to my blood. I need to move them back. And when I move them back, I'm moving them from my kidney tubules to my peritubular capillaries. So we're gonna call this step is my tubular reabsorption. So this is my glomerular filtration, and this one is my tubular reabsorption. What if you reabsorb stuff along with the stuff that you need, other stuff that you don't need or you want to get rid of? You won't settle. You will not accept it. So what you're going to need to do, you need to throw it back out in the urine. If stuff that you still have in your circulation that you failed to get filtered in the first place, you won't be keeping them. You will need to actively release them out in the urine. This third step is my tubular secretion. So again, 3 steps: glomerular filtration, tubular reabsorption, and then tubular secretion. This is a relatively important diagram. It doesn't have much details that I need you to worry about, except for the 3 steps of the urine formation. And I would rate this diagram as a 3 stars diagram. Another must be on your test diagram is a diagram showing the different components of my urinary bladder. All right, so what can we see on here? First things that we can notice is that the urinary bladder sits underneath my peritoneum. So this membrane covering that I have is not fully surrounding the urinary bladder, but instead the urinary bladder is actually sitting underneath that peritoneum that surrounds the abdominal organs, or most of the abdominal. What can we see on here? I have 2 ureters that will be traveling behind my bladder to conduct the urine through the posterior wall of the urinary bladder. And if you remember, the opening through which the urine travels from the ureters into the bladder, those are my ureteric orifices. The orifice means the opening of the ureter, so we call those are my ureteric orifices. And remember, what is the logic behind having those ureters to be connected to the posterior wall of the bladder? Is that we don't want the urine to flow back up towards the kidney. I don't want the urine to flow up back to the kidney. What I want instead, I want when I contract the bladder to squeeze the urine out, I want it to kink the ureter to act as if it's a valve. So the same action that squeezes the urine kinks the ureter along with it. And as it kinks the ureter, it prevents the backflow of the urine up towards my kidney. This urine, as it fills the bladder, is going to be funneled down through a funnel or a triangular shaped portion of the bladder. We're going to call this as my trigone. And remember, this is the whole triangle. The trigone is a whole triangle. And the apex of the trigone that connects to the urethra. You see here, this is my urethra. the apex of the trigone that meets the urethra. We're gonna call this is my here bladder neck. Please, on the exam, do not confuse the trigone with the bladder neck. So the bladder neck is just the apex, this exact point. Otherwise, it's gonna be trigone. So the whole thing here is trigone. This point that serves as the junction between the apex of the trigone and the urethra is my bladder neck. We can also see here in the bladder, before we move down, the muscle located in the wall of the urinary bladder. We call this is my detrusor muscle. So this is my detrusor muscle. It's under involuntary control. It's going to be mediated by the parasympathetic nervous system. So we don't have any voluntary control on it. It's part of the, it's going to be innervated by the autonomic nervous system. So this is my detrusor muscle. And if you see on here, when we look closer at the inside of the bladder, I see those mucosal lining ridges. So the mucosa has ridges on its surface that faces the inside of the bladder. So what would we call this? We call those are my rugae. Rugae. So the rugae is different than the detrusor muscle. Remember what is the detrusor muscle? Is all this in the wall of the bladder. But the ridges on the inside of the lining of the bladder, those gonna be my rugae. Moving on to the urethra, I see here this muscle on here. This is my internal urethral sphincter. Internal urethral sphincter. And internal, remember, internal is always involuntary. You do not have control on it. Similar to my detrusor muscle. I don't have control on it. And those are the ones that when contracted, they are not regulated by your somatic motor nervous system. They are under involuntary control. Detrusor and my internal urethral sphincter are regulated by the autonomic nervous system. To be more specific, the parasympathetic nervous system. Remember, for micturition to take place, for you to void the bladder, what needs to happen? The bladder needs to distend. And as it distends, it sends out signals to your nervous system. And the nervous system, the parasympathetic nervous system, going to respond to it by stimulating the detrusor muscle and the internal urethral sphincter. So when the detrusor muscle gets stimulated, it starts to contract. And as it contracts, it squeezes the urine. How about the internal urethral sphincter? The internal urethral sphincter, when it contracts, is going to be opening. So I open the sphincter when I contract, because this is how it looks like. Those are the fibers. So when I shorten the fibers, what's going to happen to the opening itself? It expands. So I will have an increase in the diameter of the opening as I contract the muscular fibers of the internal urethral sphincter. So does this mean micturition is totally involuntary? Remember, as we've discussed, it's partly voluntary. So when the detrusor muscle and the internal urethral sphincter contract, you feel the urge to void. But you hold. How do you hold? This is the voluntary control by contracting the external urethral sphincter. This is part of the pelvic floor, the urogenital diaphragm. So all of that all together is my urogenital diaphragm. Part of it is my external urethral sphincter. So if I put my marking on the exam, on here, and I did ask you to identify the marked structure, what do you think would you identify this as? What do you think? External urethral sphincter. External urethral sphincter. You don't mention it as urogenital diaphragm unless I'm pointing outwards, away from the sphincter itself. So here for this exam, this part is although it is part of the urogenital diaphragm, but we will not consider it because it's not the best answer. The best answer for us is going to be the most specific one. All right, so looking here at another important diagram. So remember the diagram covering the urinary bladder here. This is a must be on your test, 10 stars. Moving on to another diagram. It's slightly of lower priorities than the previous one, but still highly important. It is a diagram showing the male urethra mainly. So the new part, the newly added part to this diagram compared to the previous one is the male urethra. Remember, the urethra in females is going to be much shorter in length compared to the male urethra. All this. And because of that, if you remember, who is more prone to develop UTIs, urinary tract infections, males or females? Because of the shorter length of the urethra in females, they are more prone to develop UTIs. For the male urethra, it is divided into 3 segments. The first part of the male urethra is the part that travels within the prostate gland. So we're going to call this portion of the urethra, it is the prostatic urethra. And then the urethra leaves the prostate gland, traveling through the urogenital diaphragm, the pelvic floor. We're going to call the portion of the urethra that travels through the pelvic floors, through the urogenital diaphragm, it is my intermediate urethra, or also called membranous urethra. So we're going to call the middle portion of the urethra here, passing through the urogenital diaphragm, surrounded by the external urethral sphincter, is going to be my intermediate or membranous urethra. And then the urethra will keep going down and forwards to travel through the penis. We're going to call this part of the urethra, it is either spongy urethra or penile urethra. Then we have the opening of the urethra to the outside in either males or females is going to be called the external urethral orifice. External urethral orifice. Questions? Any questions? Please respond for me to make sure that you're listening to me, whether you do or not have any questions. So please say yes or no in the chatting box. You mean the essay question, Adib? Yes, I will review it just after this last diagram. Yes, I will be posting it in a follow-up announcement, the video recording. All right, so looking at this slide, what are we looking here at? We're looking at a histological image showing the different parts of my nephron. I've got a cluster of capillaries surrounded by an empty space around it. So this cluster of capillaries is called my glomerulus. Glomerulus. And what do we call this white space around it? This is my capsular space of the Bowman's capsule. All right, so you can simply consider this as my Bowman's capsule or the glomerular capsule, and the cluster of vessels inside, this is my glomerulus. How about those tubes, those different tubules around it, scattered all around? Those tubules are gonna be either proximal convoluted tubules or distal convoluted tubules. But how would I tell? How would I tell whether it's a proximal or a distal convoluted tubule? Have a closer look at the lumen. When you look at the lumen of the tube, if it's fuzzy, it's not clear, this tells you that you are looking at a proximal convoluted tubule. If the lumen of the tube is clear, it is going to be part of my distal convoluted tubule. So you have proximal convoluted and distal convoluted. How would you recognize proximal? It has a fuzzy lumen. And why do I have a fuzzy lumen? Because proximal convoluted tubules, remember, they are performing most of the reabsorption. 65%, if you remember, of the sodium, of the water gets reabsorbed in the proximal convoluted tubules. All your organic nutrients, all the glucose, everything gets reabsorbed here in the proximal convoluted tubules. Can you repeat that? Who did ask something? All right, so if I'm looking at here, proximal convoluted tubules will have dense microvilli. That's why their lumen is going to be fuzzy. And the distal convoluted tubules will have clear lumens because they have much fewer microvilli associated to them. Any questions? Any questions? All right, so moving on to our essay question here, for those of you who were asking about it. What is the essay question? Going to simply ask from you. I would simply ask you to describe how the kidney and the heart regulate the blood pressure based on our discussion in this module. So they are interconnected with other concepts So the heart and kidney gonna also play other roles than the ones that we've discussed in this module. So we are mainly concerned about what we've discussed in this module. So if you put this question on Google, you get a wrong answer. It will try to give you all sorts of information, but it will not give you the actual answers that we have learned. So how does it regulate? What I need you to mention here. First, you need to think kidney relies on the blood pressure. So it needs blood pressure for it to extract the urine. If I don't have blood pressure, I have a drop in the blood pressure, what's going to happen? I won't be able to function as a kidney because I rely on the pressure of the blood coming in. So the first concept I need you to keep in the back of your mind is that the kidney hates low blood pressure. And if it hates low blood pressure, what it will try to do, it needs to detect any time that you develop a low blood pressure. So this is the first concept I want you to discuss. And it wants to do what? To increase the blood pressure back to normal. So the kidney is going to be doing two main things. You can get them logically. I rely on enough blood pressure coming in for me to be able to filter that blood and extract the urine. So what would I hate the most is not getting enough blood into the kidney. So the kidney needs to know that there is no enough blood coming in, that there is a decrease in the blood pressure. And as a response to that decline in the blood pressure, it will try to increase it back to normal. This is how I will think about it. I keep this in the back of my mind all the time. How about the heart? You remember, the heart, when it pushes the blood out, it has to push it against the resistance of the previously existing blood in the vessels. In other words, it is my blood pressure. So I have here my blood pressure within my arteries, and I need, as a heart, to pump new blood out into the arteries. I need to exert extra effort. So it's like a burden. We call this burden, it's my afterload. It is the amount of pressure that the heart needs to overcome in order to pump a certain amount of blood. So this will dramatically increase if I have hypertension. So in case of hypertension, it will be difficult for me to pump the blood out. So I need to exert extra effort. If this happens, if my blood pressure is too high. So what we're looking for, what is logically can be interpreted from what we just mentioned, is that the heart hates high blood pressure. And it wants to reduce it back to normal. So first questions that we need to think of and include in our response is how I detect that you have a high blood pressure as a heart. So the heart wants to know whether you have a high blood pressure or not. And it wants to do what? To respond by reducing that blood pressure back to normal. So this is the foundational knowledge that has to be part of your thinking process. The kidney relies on having enough pressure to filter the blood to extract the urine. So it will try to detect any decline in my blood pressure and needs to respond by increasing that blood pressure back up to normal. However, the heart, having high systemic blood pressure, is going to be a burden on my heart. Why? Because the heart simply needs to exert extra force to pump the same amount of blood, this time against a higher resistance. So the blood needs to overcome that resistance to pump the blood. So you don't need to mention that directly in your essay question, but you have to keep it in the back of your mind for you to not lose track of what you're writing. So first, I need to discuss how the kidney detects that I have a, what am I trying to detect as a kidney? Can you remind me? What am I trying to detect? Low or high blood pressure? Low blood pressure. This is what I hate the most. So in case of low blood pressure, this is how I would write it down. I will put like points here as a summary for you to remember. So when you look at those points, you can recall all the information that you need to add to the question. And it's going to be, you put them in sentences to complete the answer. So here I'm detecting low blood pressure by a set of cells called my granular cells. So who detects the decline in the blood pressure? My granular cells. How? By detecting a decrease in the degree of stretch. So if you remember, the granular cells are what? Are modified smooth muscle cells lining my afferent arteriole. So the blood comes in, it's going to be pushing the wall of the vessels, applying stretch. If I'm not having enough blood coming in, I have low blood pressure, I develop less stretch on the wall of the vessels. This is what gets detected by the granular cells. This is how I detect it. How would I respond? I respond by releasing the same granular cells that did detect are the ones that will respond. How would I respond? I respond by releasing renin. So how's this renin gonna end up increasing my blood pressure? This is what will take some time to explain here. So I detected by detecting the decline in the degree of stretch associated with low blood pressure. By whom? By the granular cells. How would I respond? The granular cells release renin outside in the blood. Renin will do what? Renin converts an enzyme normally circulating in my bloodstream, coming out from my liver, called angiotensinogen. So renin will do what? Renin converts it into another form called angiotensin 1. This is not an active form of a hormone. It needs further activation. So angiotensin 1 gets converted into angiotensin 2 by an enzyme kept in my blood vessels of the lungs. The endothelial lining of the vessels of the lungs are gonna have an enzyme that converts the angiotensin 1 into angiotensin 2. So we called this enzyme that is responsible for the conversion of the angiotensin, we called it angiotensin converting enzyme, or ACE. So now I need to explain how this angiotensin gonna end up increasing my blood pressure. So what did I explain so far? I mentioned that granular cells are gonna detect the decrease in the blood pressure by detecting a decline in the amount of stretch applied to the wall of the vessel. And as a result of detecting that decline, this means I have low blood pressure. So what would I do? I release renin. Renin gonna convert angiotensinogen or activates angiotensinogen into angiotensin 1. It's not an active form yet. It will further be activated by getting converted into angiotensin 2 by an enzyme called ACE, angiotensin converting enzyme. Then I need to explain how this angiotensin 2 gonna end up getting me to what I want. What I want is an increase in the blood pressure as a kidney. In case of low blood pressure, I want to raise it back up to normal. So what will I do? As angiotensin 2 here to raise the blood pressure? Many things. 5 of them that we have discussed. First of them is going to be vasoconstriction of the arterioles. For those 5 ways by which angiotensin 2 increase the blood pressure, you don't have to describe in long sentences. You just keep it short, put it in form of points to not get confused. So vasoconstriction of the arterioles will increase my resistance. And remember, an increased resistance, an increase in the total peripheral resistance can end up increasing your blood pressure. The second effect by which angiotensin 2 gonna increase my blood pressure is gonna be by increasing the sodium reabsorption. And this can be taken place directly or indirectly. The most potent way that has the most effect is your indirect way. How's the angiotensin 2 gonna increase the pressure? VC vasoconstriction. Deep. All right. Constriction of the vessels. The vessels will constrict means they narrow. All right, so second way by which angiotensin 2 increases my blood pressure is going to be increasing the sodium reabsorption. And again, how do I increase the sodium reabsorption? Either I go by myself and tell the cells to bring in the sodium back to my blood, or I will go to the adrenal gland and stimulate the release of another hormone that is much more potent in terms of the sodium reabsorption. Is called my aldosterone. So the aldosterone release is what actually gonna be performing most of the sodium reabsorption mediated by angiotensin 2, not angiotensin 2 by itself. And when you reabsorb the sodium, remember, this increases your blood volume, and so your blood pressure goes up. So I end up here by increasing the sodium reabsorption, increasing the blood volume, and so the pressure goes up. Another way by which angiotensin II increases my blood pressure, if you remember, was by acting on two things in the hypothalamus. Hypothalamus here, part of your brain. What are the two parts in the hypothalamus that you're going to be acting on? First is the hypothalamic thirst center. It gets stimulated. And when you stimulate the thirst center in the hypothalamus, you're going to drink a beverage. And as you drink a beverage, what happens? Your blood volume increases, and so the blood pressure increases. The second way by which angiotensin II raises the blood pressure by acting on the hypothalamus is that it stimulates it to release antidiuretic hormone, ADH. Antidiuretic hormone, if you remember from the name, anti means it prevents. Diuresis means water loss in the urine. So it is a hormone that prevents water loss in the urine. So if I'm not allowing the water to be lost out in the urine, I am retaining that water. So that builds up as an increase in my blood volume, and so an increase in my blood pressure, which will end up increasing my filtration. And this is what I want here. You need to have enough blood pressure coming in order to filter that blood. Next is going to be the constriction of my efferent arterioles. You remember how the constriction of the efferent arteriole going to play a role in here? The blood gets into my glomerulus through the afferent arteriole. This is my glomerulus. And the remaining of the blood that won't get filtered gonna be moving out from the glomerulus along my efferent arteriole. Remember what's gonna happen if I constricted my efferent arteriole. Remember, those are my proximal convoluted tubules, distal convoluted, all the rest of the tubules, doesn't matter. Where the blood goes next, as it travels through the efferent arteriole, it will be directed around my kidney tubules as my peritubular capillaries. So as I constrict the efferent arteriole, apply pressure here, reduce the diameter of the vessel, it gets narrowed, so less blood gonna be able to travel here to my peritubular capillaries. And if less blood moves in here, the pressure of the blood inside my peritubular capillaries gonna decline. Or the hydrostatic pressure of the blood in my peritubular capillaries will decrease. So if I have low pressure in my peritubular capillaries, and I have, on the other hand, high pressure, high hydrostatic pressure in the filtrate, what's gonna happen? If I have a pressure difference, the water gonna be able to move back to my blood. From the kidney tubules back to your blood. Why again? Because here you have higher pressure compared to here you have lower pressure. So this constriction of the efferent arterioles is gonna end up developing an increase in the reabsorption. And if I have an increase in the reabsorption, what's gonna happen? My blood volume increases, and so my blood pressure will go up. All right, so what do I need to mention all the details on here? No. On the exam, in your essay question, you don't have to mention all the details, but I explain it for it to make sense, for you to remember it. But you don't have to mention all the details on here. You're gonna just mention that angiotensin II is enhancing the constriction of the efferent arterioles, which increases the water reabsorption, and so blood pressure goes up. Does this make sense so far? Any questions? Any questions? All right. So the last way by which angiotensin II, as we've discussed, increases my blood pressure is going to be by contracting the extraglomerular mesangial cells. All right, so if I'm looking on here, this is my glomerular capillaries. Like this. And this is my Bowman's capsule. And the extraglomerular mesangial cells are sitting here, next to the glomerulars. Those are the ones I want to contract here in response to the angiotensin II. Remember, in order for anything to move from the vessels to your Bowman's capsule, I need to have a contact point between the vessel and the capsule. Otherwise, nothing gonna pass. So the vessel needs to be touching the tube. So what is the area available for the fluid to pass through from the vessels to my Bowman's capsule? All the areas where they touch one another. So all of that is gonna be a surface area available for filtration to take place through. What happens when I contract those extraglomerular mesangial cells up in here? I will end up pulling the vessels away from the Bowman's capsule. So the contact area between the vessel and my Bowman's capsule has been dramatically reduced. So only surface area going to be available is this segment on here, where they touch one another. Otherwise, all this here has been lost. It's not an available surface area for filtration to take place through. So what's going to happen if I'm not filtering as much as before? If I was supposed to filter 1 liter, now I filtered half liter. So this means I have kept the other half. So I was supposed to spend whatever, like $1,000, and instead I spent 500. So as if I've gained 500. The difference between what I was expecting to spend and what I actually spent. If I spent less than what I was expecting, so this means I've saved the difference. So this is the same concept. So if I didn't allow this fluid to get out, because I have reduced the contact surface between the vessels and the woman's capsule, this is going to tell me that I have gained volume. How did you gain volume? You didn't add any new water. You simply didn't lose what you were expecting to lose. So if you have lost less of what you were expecting to lose, this makes you as if you have gained the difference. So contraction. So what you need, you don't need to be drawing all that. You don't have to explain all that. What I need you to remember is that in your response to add that concept, that angiotensin 2 gonna contract the extraglomerular mesangial cells, and as a result, you don't have to explain the details on here. And as a result of that contraction, I have a reduction in my filtration. This is what I need you to remember. Does this make sense so far? Any questions? Any questions? All right, so we can have a break for 10. We come back and we're going to be answering the second half of the essay question on here, which is the response of the heart by which it will be reducing the blood pressure in case of a abnormally higher blood pressure. All good? All good so far? So we will have a break for 10. We will come back to complete our discussion of this essay question. Can you hear me now? No? Yes? All right, great. All right, so let's discuss this remaining part of the essay question. So it is so simple. If you understand what we have previously discussed, it's so easy to mimic it in a reverse way to describe how the heart gonna be reducing the blood pressure. So first question that we have in mind is how does the heart detect that you have a high blood pressure? So the heart doesn't have receptors up front to figure out what is the pressure deep into the arteries. It figures out that you have high blood pressure by detecting the increase in the amount of blood coming back to it. So if I have a high blood pressure, the pressure in the whole circulatory system is going to be increased. So as my veins. So the amount of blood that is going to be driven back towards the heart is going to go up. This is how the heart detects that you have a high blood pressure. So if you have high blood pressure, the venous return going to increase. And as the venous return increases, this will increase the degree of stretch applied to the wall of the atrium. So more blood coming in, atria will distend. So I have, as a result, an increase in the degree of stretch applied to the wall of the atrium. So similar way, we have figured out that we've got a reduction in the blood pressure by detecting a decline in the stretch. But here, as we have high blood pressure, we get to detect an increase in the degree of stretch. So if I increase the degree of stretch of the atrium, the atria will respond by releasing a hormone called atrial natriuretic peptide, or ANP. And this is fine for us to use ANP. It's scientifically accepted. It's a scientific abbreviation. So you don't have to write down the whole name. ANP targets 3 organs. Each of them is releasing a hormone that increases my blood pressure. So what will I do? Simply speaking, I affect 3 organs and inhibit them from releasing their hormones. So what is the first organ? It's going to be my kidney. To be more specific, the granular cells. What were the granular cells releasing that increased my blood pressure? As we've mentioned earlier, they were releasing renin. So if I want to reduce the blood pressure as ANP, what I will do for the granular cells, I inhibit the release of renin. And if I don't have renin, you remember, would I be able to get angiotensin II? No. So you relied on the conversion of the angiotensinogen into angiotensin I in order to end up developing angiotensin II. So renin is critical for you to end up having angiotensin II. And the very first effect of angiotensin II that we've mentioned is that it constricts the blood vessels. So if I don't have renin, I will not develop angiotensin II. And if I don't have angiotensin II, I will not constrict the vessels. So the vessels will dilate. I will end up developing a vasodilation, which causes a reduction in the blood pressure. And this is what is the target that the heart is trying to reach, is a reduction in the blood pressure whenever it goes up. The second target organ for ANP is your hypothalamus and the posterior pituitary gland. And if you remember, the hormone that was being released from my hypothalamus was ADH. So what am I trying to do here, affecting the hypothalamus and posterior pituitary gland, is to inhibit the release of ADH. So the target organ, let me write it, maintain consistency here. So my target organ is the hypothalamus, and on which I inhibit the release of ADH. If you remember, what was the function of ADH? Prevents you from losing the water out in the urine. So my function as ADH was to pull water back to my blood. If I don't have ADH, what's going to happen to this water is going to fail to be reabsorbed. So I won't have reabsorption of water. So this water going to leave, causing a decline in my blood volume that results in a decrease in my blood pressure. and this is my target. This is what I want. The third effect of the ANP is reached by acting on the adrenal cortex. And if you remember, as we mentioned earlier, what was the hormone that increases the blood pressure coming from the adrenal cortex is my aldosterone. And what was the function of the aldosterone? It was to increase my sodium reabsorption. And as I pull the sodium back, it's going to be followed with water, increasing my blood volume and so blood pressure. So if I want to reduce the blood pressure, as ANP, it acts on the adrenal cortex to inhibit the release of aldosterone. And when I inhibit the release of aldosterone, I have a reduction in the sodium and water reabsorption. And if I'm not reabsorbing water and sodium, this is going to decrease my blood volume. And as I decrease the blood volume, I end up having a lower blood pressure. So this is simply what you need to mention in your essay question. One quick recap: kidney responds to a decrease in the blood pressure by increasing it. How does it detect that there is a decline in your blood pressure? By detecting the decrease in the degree of stretch applied to my granular cells. And in response to the degrees of stretch, the granular cells release renin that converts angiotensinogen into angiotensin 1, that will then be converted into angiotensin 2. Angiotensin 2 acts by different means to reduce your blood pressure: constriction of the arterioles, which raises the resistance and so pressure; increasing the sodium reabsorption directly and indirectly by stimulating the adrenal cortex to release aldosterone. Angiotensin 2 acts on the hypothalamus, stimulating the hypothalamic thirst center for you to drink a beverage. And so you can absorb this water, increasing your blood volume back to normal, and stimulating the hypothalamic thirst center to release ADH that prevents you from losing the water out in the urine. Retention of this water can end up causing an increase in your blood volume, and so your blood pressure goes up. Another action of angiotensin 2 was the constriction of the efferent arteriole. And if I constricted the efferent arteriole, remember, the blood traveling in your peritubular capillaries is going to decrease. And if I decrease the blood traveling in my peritubular capillaries, the gap between the fluid pressure in my tubules and in my peritubular capillaries increases. So as a result, the fluid is going to be moving back to my blood, increasing my water reabsorption. Last mechanism of action for angiotensin 2 was the contraction of the extraglomerular mesangial cells. And as I contract the extraglomerular mesangial cells, I'm pulling the vessels away from my Bowman's capsule. So the contact surface area between the vessel and the Bowman's capsule is going to be reduced. And as a result, I have reduced the filtration. I'm not losing the fluid out, it's retained as blood volume, and so it will be increasing my blood pressure. How the heart responds in case of a state of high blood pressure? Remember, the heart detects that you've got a high blood pressure by detecting the venous return. How does it detect the venous return? The more blood you're getting back means you have higher blood pressure. And this can be detected by detecting the degree of stretch applied to the wall of the atrium. If the atria did stretch, this means I have a high blood pressure and I need to respond. How do I respond as atria? I release atrial natriuretic peptide, or ANP. ANP targets 3 organs, inhibiting 3 hormones. Acting on the kidney and the granular cells, I'm inhibiting the release of renin. If I don't have renin, I won't get angiotensin II, and if I don't have angiotensin II, my blood vessels will not constrict, but they will dilate. No deducted points for grammar, no. The second target organ here for my ANP is by inhibiting the hypothalamus from releasing ADH. And if I don't have ADH released from the hypothalamus, this will prevent me from getting the water back to my blood. So I have a reduction in the blood volume, and so a reduction in my blood pressure. ANP also acts on my adrenal cortex, inhibiting the production of aldosterone. Aldosterone normally reabsorbs the sodium back to your blood, and it's going to be followed by water. So if you inhibit the reabsorption of the sodium, water will not follow, and so you're going to get rid of that water out in the urine, declining your blood volume, and so you get a decline in your blood pressure. All right, is this all clear? I won't be discussing any new material. I want you to focus on studying this weekend for your upcoming exam. So please, when you are studying for the lecture exam, your first priority is your quizzes. The current graded quizzes is your first priority. So please make sure that you go over the slides, try to answer as many questions as possible from your Q banks, and then take your quiz. Not visible body, no. Visible body is going to be a lab assignment. No. The most important and your first priority is your actual quizzes on campus. Your lecture exam is going to be covering the same concepts addressed in your quizzes. So please focus on those ones for your lecture exam. And we have today covered everything that you need to know about for the lab exam. All right, it's not uncommon to have 100% on those 2 exams. So the other AP2 class that I'm teaching currently, they have I believe 10 or 12 people who already got 100% on this test. So it's not hard to get 100% on this exam. Any questions for me? Any questions? All right, so thank you so much for making it to the meeting today, and we'll see you all back in class on Tuesday. Enjoy your weekend.